Yeah, Gitanjali.
Okay, then we will start. Okay. So, welcome to the second session of our Journal Club of Omics Bootcamp. Today we have two speakers. Our first one is Joshua Stephen, and his topic is time is valuable. I mean, of course, time is valuable, but why the time is valuable? Let find. I mean, Joshua will help us to find that. So let's start. And one more thing, those who want to ask question, please give your question in chat box with your mail ID, so that if not possible now, we can send your answer. So let's just fasten your seat belts and start our journey. I mean time journey. Okay, let's start.
All right, all the best. Start the show.
Okay, shall I start presenting?
No, you are not
Yes, please
presenting
start.
anything. Okay. Yeah.
Hello, can you see my slides?
Yes. Yes, we can see your slides, Mr. Joshua.
Okay, ma'am, I'll start. Good afternoon. I'm Joshua Stephen, doing my second year MSc Biotechnology in Bharatiyar University. And this is the second Omics Bootcamp, and I'm going to talk about the tumor immune microenvironment of non-small cell lung cancer. The topic is time is valuable, understanding the dynamics of tumor immune microenvironment in non-small cell lung cancer. I'd like to thank my mentor, Dr. Seema Shah, Assistant Professor of Biochemistry, AIMS, Raipur. So these are the things that we are going to see today. So, introduction to non-small cell lung cancer. Lung cancer is the most prevalent cancer in the world until because last year breast cancer, as you can see in this pie chart, has taken lung cancer, and breast cancer accounts for totally 11.7% of cancers, while lung cancer accounts for 11.4% of cancers. But still, the number of deaths caused by lung cancer is higher than any other cancer, as you can see from the second figure. These are the statistics from last year, number of deaths for each cancer, and lung cancer surpasses all other cancers. So, lung cancer can be broadly categorized by the microscopically as small cell lung cancer, which accounts for 15%, and a non-small cell lung cancer, which accounts for 85% of lung cancers. The subtypes of non-small cell lung cancer are adenocarcinoma, squamous cell carcinoma, and large cell carcinoma. And non-small cell lung cancer, though it is most prevalent, is also the most studied cancer in medical oncology and pulmonology. But are the efforts sufficient? No, because most patients are still diagnosed at a very advanced stage. It's usually stage 3 or stage 4, and the 5-year survival rate is still around 15 to 25%, which is very low, and it still causes the most number of deaths among cancer. So let's see about the clinically significant phenomena in non-small cell lung cancer. First one is epithelial to mesenchymal transition. So this is the transition wherein the tumor cell starts de-differentiating and it attains stem-like properties. It loses its cell-cell addition and polarity and gains migratory and stem cell-like properties. So most of the time it promotes metastasis. In some cases, it actually makes the migratory cell more prone to be surveyed by the immune system. So those are exceptions. But most of the time, it helps metastasis. The second phenomenon is tyrosine kinase inhibitor resistance. So one of the main pathogenic root of non-small cell lung cancer is an activating mutation in EGFR protein. So the epidermal growth factor receptor is the cell surface receptor that binds to the growth factor and activates the pathway that eventually leads to the proliferation of the cell. So you can imagine that if this receptor is activated constitutively, it will signal the cell to proliferate even in the absence of a growth factor. So that's what exactly happens in about a third of non-small cell lung cancers, where different mutations in EGFR makes it constitutively active. But thankfully, we have come against this therapeutically using different antibodies, where we target these mutations and we call them tyrosine kinase inhibitors, because EGFR is a type of tyrosine kinase, receptor tyrosine kinase. But the thing is that due to clonal propagation, more and more mutations keep accumulating in the EGFR, and the old mutations that we target, they get hidden by new mutations. So we have gone from, as you can see, first generation TKIs and second generation TKIs and third generation TKIs, and the cancer has been showing resistance in all levels. So we're studying fourth generation TKIs too. So metastasis, it's a problem with all cancers. In non-small cell lung cancer, it's even more problematic. And the main sites are brain, liver, bone, and brain metastasis alone account for 30 to 40% of the metastasis. And due to metastasis, 70% of the deaths happen in non-small cell lung cancer. So the tumor immune microenvironment, that's the major topic today. And we all know about the tumor microenvironment, where we consider all the immune components as well as the non-immune components of the microenvironment. But the immune components alone are extremely diverse. And in the past decade, so many studies have been carried out and they show the highly dynamic and diverse nature of the immune component of the microenvironment. And that's why studies have slowly started adapting the term tumor immune microenvironment. It's still a relatively new term, and it's necessary since we need to zoom in and focus more on this immune component of the microenvironment. So that's what time is about. These pictures are from a review which is exclusively on time, and they have even tried to classify time, and we can see three subcategories as infiltrated, excluded, where certain cells are included, infiltrated, and certain tumor infiltrating lymphocytes, they are actually excluded, they are in the periphery. And here we can see infiltrated and inflamed. Inflamed in the sense that the secretum of the immune cells consists more of inflammatory cytokines. And we can see infiltrated and TLS. This is when tertiary lymphoid structures start to begin when the tumor cells, immune cells itself, they start like proliferating more than necessary. So various correlations between different time components, different time signatures, and non-small cell lung cancer progression have been established. So we can see how this can be useful for us. So major phenomena behind time dynamics. So exclusion of certain immune cells. So CD4, CD8 T cells are excluded in different subclasses of non-small cell lung cancer. This is in this example, it's squamous cell carcinoma. During EM, cells are excluded. And enrichment of certain other immune cells. For example, Th17 cells are often accumulated in non-small cell lung cancer because they produce IL-17, which is pro-inflammatory and most of the time pro-tumor in nature. So, and different expression of immune checkpoint proteins. Different immune checkpoint proteins like PD-L1, PD-1, and CTLA4 are often overexpressed in tumors. So these checkpoints, they can bind with receptors, complementary proteins in the immune cells, and the binding activates the apoptosis and thereby suppresses the immune response to the tumor. So we can target these immune checkpoint proteins as well, and that's called immune checkpoint blockade. And underexpression of cytokines, so IL-37 is a well-known anti-tumor cytokines, and it is underexpressed in many non-small cell lung cancers. And overexpression of pro-tumor and pro-inflammatory cytokines like IL-17. So we'll see two tables here that I curated using results from different studies. So we can see how the time changes with different, the three main phenomena that we saw: EMT, metastasis, and tyrosine kinase inhibitor resistance. So CD8 cells, they are excluded when EMT happens, but we see more infiltration when metastasis happens. So that means they are aiding in a way. They become mutated and start producing proteomic cytokines in certain cases. So like this, we can see a whole lot of signatures. This is pretty basic. When we go deeper, it gets really more confusing because multivariate here. So here's a small that we can understand how time is useful for non-small cell lung cancer prognosis. So here, when in the immune component, when the high CD103 plus tumor infiltrating lymphocytes are there in squamous cell carcinoma, increased progression-free survival as well as overall survival has been noticed. And during elevated neutrophil lymphocyte ratio prior to nivolumab treatment, immune checkpoint blockade, in recurrent metastatic non-small cell lung cancer, it's associated with poor outcomes. So just like that, we can see a lot of associations of different time signatures in different non-small cell lung subtypes and the prognosis that we have for them. Good timers. Need for omics. So that's where we come to where omics actually helps in the study of time. Here we review two studies, two independent studies that actually try to correlate immune cells and cancer stem cells. Cancer stem cells, as you know, are like they are more like stem cells, not de-differentiated cancer cells, as you can see. So they resistance to chemotherapy, radiotherapy, and a whole lot of therapies. Immune cells, on the other hand, they are against the tumor most of the time, except for in cases like Th17 immune cells. These studies, both of them try to correlate uh, cancer stem cells and tumor infiltrating lymphocytes in non-small cell lung cancer. This first study, it used ALDH as a marker for and CD3, CD4, and CD8 markers for immune cells. And they found a positive correlation between these cancer stem cells and CD8 lymphocytes, and also cancer stem cells and CD3 cells. So that's very intriguing because we know that cancer cells, stem cells, they promote the tumor. While CD8 plus CD3 plus cells, most of the time, if not all of the time, they're against the tumors. So this is very intriguing. So another study, they also had a similar goal and they used OPT4 and CD123. They are other markers for cancer stem cells. And for immune cells, they used CD8 and CD56 as the markers. So CD8 is again TC cell and CD56 it is NK cell, it's also anti-tumor. So they found a negative correlation between these immune cells and cancer stem cells. So we expect a negative correlation, but this study has given a negative correlation while this one has given a positive correlation. And the thing is that both of them are quite recent and are done in the omics era. But the thing is that omics is more suited for high throughput screening where we can take a holistic approach and consider a whole lot of markers. But they haven't done that here. They have used ALDH here for assaying, and here they have used OP4 and CD3 via FACS to quantify the cancer stem cells. So the statement is, could this confusion be avoided if they had used multiple markers for tumor infiltrating lymphocytes and cancer stem cells? So omics for understanding time dynamics. Since time is persisting with reductionist approaches, the previous example would be a good example of a reductionist approach where they dissected and chose just one marker. And how reductionist approaches will not only be less capable of answering our questions, but will in fact cause new unnecessary questions as well. Omics technologies are by nature suited to study diverse systems and are hence highly utilizable tools for studying the time. So let's have a brief run through of a few selected articles that show the use of omics in studying time. This is the first one. The title is DNA methylation profiling of non-small cell lung cancer reveals a COPD-driven immune-related signature. Okay, COPD is chronic obstructive pulmonary disease. So let's see the methods. DNA isolation was done, followed by methylation profiling. and RNA isolation was done, followed by transcriptome sequencing, and histology and immunohistochemistry was done to see the tumor infiltrating lymphocytes and the dynamics of that. And and a lot of statistical analysis was done to cluster the samples, and validation for all the results were done independently using data collected from the Kansas Genome Atlas. So what they found is that first, in clustering, they clustered the whole set of samples and obtained two clusters. And one of the clusters was highly associated with COPD. In this cluster, which is associated with COPD, genes in the tumor tissue were hypermethylated. So we know that when a gene is methylated in the promoter, it means it will be underexpressed. So they also saw the transcriptome, which is the expression, and they also found that it was underexpressed. They were hypermethylated in the promoter, CPGs, and they were also underexpressed. And these genes were found to be immune-related genes. The immune signature was attributed to both differential methylation of immune genes in the tumor and the differential immune cell tumor infiltration, which was found by histology and immunohistochemistry. So, what is the application of this? COPD specifically affects the tumor methylation profile more than other expected factors such as age, emphysema, smoking, etc. Smoking, it's a high risk factor for both small cell and non-small cell lung cancer, but COPD is more strongly associated with this immune signature. So this gives an explanation of the already known dysfunctional immunity in COPD non-small cell lung cancer patients. So we already know that immunity is compromised and it is dysfunctional in COPD patients having non-small cell lung cancer. So this gives a reason for that. And this establishment helps to direct more exploratory national studies to better treat COPD non-small cell lung cancer patients. Because now that we have identified these changes in the immune signature, we can try to counter these changes. So let's have a look at two figures from the studies. This is the methylation analysis. So we have 12 immune-related genes here, and in the y-axis we have the expression. No, we have the methylation. So we have two box plots for each of the genes. The gray plot is the tumor, and the white one is the adjacent non-tumor tissue. We can see that for all the genes, the tumor has more methylation. So when they came across this, they thought the expression will be less for all these tumor genes. And as it happens, here is the expression data. And here all the gray boxes, which are the tumor tissues, are underexpressed compared to the adjacent tissues for all the immune genes. So these genes are underexpressed in COPD driven non-small cell lung cancer. So now that we have established this, we can take a lot of translational research and can address these issues. So here, transcriptome and epigenomics has helped methylation profiling and transcriptomes. So next, proteomic identification of exosomal LRG1, leucine-rich glycoprotein 1, a potential urinary biomarker for detecting non-small cell lung cancer. Methods: urine sample collection, isolation of exosomes by ultracentrifugation, and negative staining, electron microscopy, and immunoelectron microscopy to validate whether the sample isolate contains the exosomes. And STS page was done, followed by in-cell digestion of a certain range of the bands. And this was taken for LC-MSMS analysis for identification, and that data was used for protein identification. And immunohistochemistry staining was also done. And findings: the band distributions in STS page are different between urinary and exosomal proteome and normal and non-small cell lung cancer samples. So that is, if you collect the urine and run it on STS page, and if you collect the urine and isolate the exosomes and run it on STS page, both are very different. And non-small cell lung cancer samples and normal samples, they are also very different. And in this difference, band intensities between 35 to 45 kilodalton were in non-small cell lung cancer samples. So genes were identified in this range. Out of this 18, 11 were unique to non-small cell lung cancers. And out of this 11, LRG1 was selected for further analysis. And Western blotting was done to quantify it, and it showed that LRG1 was overexpressed in non-small cell lung cancer. And then we have to know whether the overexpression is specific to the tumor tissue alone or if it's seen in the adjacent tissue as well. So they did immunohistochemistry and found that the overexpression of LRG1 is specific to only the tumor tissue. So you can see in the small figure from the study that these are the cancer samples and these are the normal samples, and these are the expression levels of LRG1 and how LRG1 is highly expressed in non-small cell lung cancer. So the application is that increased LRG1 in urinary exosomes might be directly derived from non-small cell lung cancer tissue, and this can be used for non-invasive diagnosis as well as prognosis. So you are supposed to spot something here because this is not much associated with tumor immune microenvironment, which is what we're talking about today. So why did I choose this paper? So I wanted to make a point that to completely understand the tumor immune microenvironment, we should not only stop with studying the immune component of the microenvironment, but we should start studying all the other things as well. In this case, LRG1, which is not a exclusive component of the immune system. And LRG1, it actually is involved in a lot of malformed blood vessels in cancers, so that the blood vessels in the cancer become really leaky, and the serum doesn't reach the whole tumor, especially the inside parts of the tumor. So in the hypoxic serum-free condition deep inside the tumor, that environment, it helps the promotion of proliferation of cancer stem cells. And cancer stem cells are, they are bad news. They promote resistance against so many therapies. So you can see how LRG1 can actually affect the distribution of different cells, and along with certain other proteins which are specific for specific antigens on like CD4 or CD8, LRG1 can actually play a role in immunoexclusion. It can be part of a new mechanisms of immuno exclusion that we still don't know. So that's so important to analyze not only the immune components but the other components and correlating it with integrating it with our understanding of the immune microenvironment. So next, it's an review article about the proteomic based approaches for the study of cytokines in lung cancer. These approaches can be used for a lot of different samples, but here it's cytokines. And quantifying cytokines is also very important since there are so many different cytokines. We saw how IL-34 is against tumor and IL-17 is pro-tumor in nature. And gel-based techniques. Gel-based techniques are 1D and 2D ST space and dyes, etc. Dyes is difference in cell electrophoresis because in ST space to compare two samples, we will do two gels and different artifacts may be involved and they will affect our results. So in dyes, we can run multiple samples with different dyes, fluorescent dyes in the same gel, so it will avoid artifacts. So merits is it's less costly and easy to perform because most of you would have performed gel-based proteomic technique already because it's so easy to perform and it's simple. Demerits is that it's low throughput, low sensitivity, and needs larger amount of samples and is time consuming. Gel-free techniques, these can be labeled or non-labeled. Labeled, they are ICHAC, SILAC, and ICAT, and non-labeled are LCMS antibody arrays. Merits, they're more sensitive. We can work at really less small sample volumes. And demerits are that they are more costly, more complex to perform, and might not be able to differentiate between certain protein isoforms if we are going to identify the whole protein using a few peptides, then we might confuse certain isoforms to be the same protein. So mass spectrometry is like the gold standard for proteomics. We use them for both gel-based and gel-free techniques. And it is widely used for protein and peptide sequencing and identification by measuring the molecular weights and charges of their ions. And various combinations of ionization methods and mass analysis, even double mass analysis we can use. And that means a lot of versatility. We can customize it according to our needs and according to our samples. Applications of studying time. So targeting and manipulating the selectively enriched immune cells in the time. Like Th17 cells, they are enriched, but we can manipulate the immune microenvironment using gene therapy or so many different things. And we can actually manipulate the same enriched cells to attack the tumors. And that is very much possible. That's what is being studied now presently. And studying the precise mechanisms of immunoexclusion will help to stop or reverse it. There are so many unknown mechanisms of immunoexclusion, I believe, and one is that the overexpression of PD1, PDL1 immune checkpoint proteins, which will suppress certain kinds of immune cells. It won't, so that will cause an exclusion, right? So we can target we are targeting by immune checkpoint blockade, but we see a lot of crosstalk. So when we actually target the immune checkpoint proteins, a new clone starts to propagate, and it will have more of EGFR mutations and less of immune checkpoint proteins. So a lot of crosstalk is involved, and that's why we need a better understanding and better prognosis. We saw in the table how using time alone we can have different kinds of prognosis. So the more understanding we have of the time, the better prognosis we can make. Avoiding immune checkpoint blockade and the prognosis for it based on the time is bad. Yes. And better therapeutic strategies. Choosing TKI and ICB in cases where TKI treatment has shown to increase immune checkpoint protein expression. This is where, like, when a tumor has EGFR mutations, we choose to go with TKI therapy, which is totally normal, understandable, and that's the right way to go. But these tumors, some of these tumors, they suddenly start overexpressing immune checkpoint proteins. That is, when we target from one route, it moves to another route. So we can actually notice certain time signatures where this happens. And when we come across this signature again, we can opt for TKI plus ICB therapy so that we can corner the tumor. So, conclusion. We're at the stage in this subdomain of time where the more we study about time, the more questions arise. And but we're also at the stage where omics technologies have become quite diverse and widespread for us to explore it. Given the available technological potential and the spectacular prospect of revolutionizing prognosis and therapy of not just non-small cell lung cancer, but also other cancers and other disease, we can expect studies about time to all but increase in this decade. These are the references that I used. Thank you for listening. You can ask your questions.
I think there are a couple of questions for you, Joshua. Just let's check the chat box. Two questions, I think there.
Yes. Okay. COPD. Okay. Yes. May I know what's the first question? Is the COPD is the first question?
No, no. Before that, there is a question. I think. Wait.
What are the applications of COPD?
Abhishek has yes. What are the most favorable prognostic factor for patients with metastatic non-small cell lung cancer?
Okay, let me
Most
see.
favorable prognostic factor for patients with metastatic non-small cell lung cancer.
Okay, most favorable prognostic factors. So in my table that I correlated, I have referenced a couple of studies that actually found that CD4 cells and CD8 cells were actually highly enriched more than normal when during metastasis. So that's actually a bad prognosis. So most favorable will be obviously in that aspect where CD4 and CD8 cells are concerned, not their enrichment, too much enrichment, and not their exclusion. The normal signature of CD4 and CD8 cells, and they are a good prognosis for metastasis where those cells are concerned. And there are so many other factors also involved, like so many immune and non-immune factors, like so many keratins, and they are also involved. Where CD8, CD4 cells are concerned, this is my view. This is their view. The result.
Okay. Another is what are the applications of COPD? I think techniques. It not techniques. It actually disorders. I think. Okay.
Yeah,
What are the
COPD.
application of
Yes.
COPD disorders?
Applications. No, COPD is a disease. It's a chronic obstructive
I also
disease.
mentioned that.
It's not a technique. I think there was a misunderstanding. So
Yes.
the application of that study, if you are asking that, the application is that we now know that immune signature of COPD patients also have non-small cell lung cancer. So we know how we know the genes which are underexpressed, So we can use different methods like gene therapy, so many different methods, or we can stop the hypermethylation too, because there are different modulators and methyl transferases. We can target them and we can stop this at the level of hypermethylation. We can change this at the level of expression, or we can just go for gene therapy. There are so many techniques to counter this under expression of immune genes in COPD non-small cell lung cancer. So we have established it, the fact. So now we have to take translational approaches.
Joshua, this is actually a very nice presentation.
Thank you, sir.
And also, I think, some of these presentation can also bring idea to all the participants here that how, more advancement in the omics technologies could contribute to, such really, tough problems of these type of cancer, like, lung cancer is still one of the most challenging one to work with, right? And especially the metastatic cases are, nobody can actually solve this as of now. When somebody has metastatic lung cancer, you feel like, now 3 months to 6 months time is left, right? So, in which way these technologies can contribute is definitely, worth thinking about. And in fact, from these group of the students, you can always think about taking some data sets from the literature or the public repositories, looking into these kind of data, and some, small projects like that could be developed from this area, and then if you come up with some hypothesis, a certain thing will be happy to help you take forward.
Yes,
So
sir.
that's what I
Great.
just thought to add from my side. Uh, of course, there could be some other technical question, but given the time which we have, I'll just leave you with one thought that in your one slide you showed an overview of different techniques being used for gel-free, gel-based mass spec, which is being used for this research. It's good idea for
Yes,
you
sir.
to show the references that using this technique, what kind of research was done, and what is the reference being used for that. That will be
Okay, sir.
helpful for the future people to follow up on these studies. And
Okay, sir.
as I've also mentioned earlier, I think we should make some mechanism that presenters should be able to share their PDF file of their PPT as well as the references, 4 or 5 key references used in the presentation.
Yes, sir.
And let's not keep any confidential information there so that anybody can access that. And that we should make a common mechanism. So I think that's all right now, I think for you, Joshua. So
Yes, sir.
uh, I think we can
So
proceed,
I can
Geetanjali.